And so I think this slide will be very common in the next few years with people talking about COVID-19, but obviously we're in the midst of a pandemic, and this pandemic is of this COVID-19 is listed by the SARS-CoV-2 virus. This coronavirus that originally harbored in bats, and like all the bats, all the epidemic or pandemics of the coronavirus, they all started in bats, and they had an intermediate host before they jumped to humans. In the case of SARS-CoV-1, which is an epidemic in China, the intermediate host was thought to be civets. In the case of MERS, which was an epidemic in the Middle East, that thought was to be camels. And in the case of SARS-CoV-2, the thought, although more evidence needs to come out, is to be pangolins. But how patients present with COVID-19, the list keeps growing at this point, but it's pretty consistent that a majority of these patients present with fever, cough, fatigue, muscle aches, and then the list goes on and on with loss of smell or nausea. And now if you look at it, the list just keeps growing. But I think one of the things that we're seeing, and was touched upon by the last two speakers, is what is the immune response? What's happening with these patients with this infection? Because in some cases, what we see is that we see a suboptimal immune response, and that is caused by the virus itself, and that's the virus disseminating throughout the tissue. And it just tells us that the immune response isn't functioning properly. So what are the cues to get it back to being normal? On the other hand, what we see is an overactive immune response. And so this is the immune system essentially going crazy, perhaps the response is too late. And what happens is you get cell types activated that are not necessarily involved with clearance of the virus, but are damaging the tissues. So in the case of COVID-19, we just don't know where we are on the spectrum. And the goal of this study was to see where majority of these patients lie. And so some of the outstanding questions we wanted to address are, what types of immune responses exist in COVID-19? Are there common pathways to immune activation? And then I think the big take-home that we're trying to say is, how does it relate to clinical features? Because if we're trying to translate our work to the clinic, it's not necessarily meaningful if we just present increases in T cell frequency or increases in B cell frequencies. We need to relate this to clinical metadata or clinical data. And so that was the last objective of this work, to see how these changes immunologically have changes clinically. And so this work was a huge effort, and it was in a three-part work with Noel Meyer, who's a pulmonary pulmonary clinician here who studies sepsis. However, when we started getting COVID-19 patients, she quickly adapted her protocol so we can start drawing these patients and studying them as soon as they presented in the ICU. This was also in work with Mike Betts and their two postdocs, Letty and Marina and Letty, who also did a lot of work in studying the non-B cells and non-T cells, and then also work from our lab, who with myself, with Josephine, Amy, Ali, Jen, Cecile, and Derek, we all kind of took on this project, and it was really a wonderful collaborative effort. And then, so how the pipeline worked is that when patients present to the clinics, we were able to just essentially draw their blood when they presented, and we also got longitudinal data at 7 or 28 days if they remained in the clinic, and we were able to then do profiling right off the fresh blood. And so we got a whole host of patients, 100. So I will show you clinical metadata on the top portion, which includes 149 patients, COVID-19 patients, 46 recovered patients, and 70 healthy donors. But I will also show you flow cytometry data on 125. So a majority of those patients we have clinical metadata for. I will also present flow data for. Throughout the talk, there's going to be a universal theme that the COVID patients are going to be highlighted in red, the recovered in blue, and the healthy donors in green. So now if you look at the age distribution, now certainly there we see some overlap in the age, but primarily a lot of COVID-19 with a median age of 60. This is consistent with what we see across the board, in that disease severity is associated with age, and so that's what we certainly see in our data set as well. And so when we look on the left here, this is looking at some of the clinical data that are collected from the ICU, and we can look at clinical measurements of inflammation as measured by ferritin D dimer or hsCRP. And the green boxes represent where the healthy donor range is. And consistent what we see in the literature, in COVID-19 patients, you have elevated ferritin, elevated D dimer, and elevated CRP levels. Now, when we look at the whole blood cell count, we don't really see much of a change of the COVID-19 to healthy donors, but when we look at the total lymphocyte count, it is certainly reduced. And this tells us there's lymphopenia in these patients, again, consistent with what we see in the literature. We can also look at the monocyte counts, and we don't see much of a difference. Now we can take all of that clinical metadata and say how to associate it. So I'm going to talk you through this one a little bit because this similar plot is going to be reoccurring. And so basically what it's saying is that it's comparing two different features, and the more red it is, the more positively correlated it is. The more blue it is, the more negative correlated it is. If there are stars in the box, that means there's significance associated with it, and the more stars means more significance. And because you're doing multiple comparisons, we need a false discovery rate. So if you see a box that's highlighted, that means it's below the false discovery rate threshold of 0.05. So something like, so how I would interpret this is males typically have stronger, higher ferritin levels, would be how to interpret this. And so one interpretation you can see is that when you present with higher disease severity in the clinic, you typically you typically are sick. So if you come in at day 7 with a high disease severity, it's likely that you'll remain sick 14 or 28 days out. And so what we can do with this type of data set is we can compare a bunch of different clinical metadata to each other to say how this disease is presenting in the clinic. And so from this, obviously as immunologists, we want to do a further look into the lymphocytes, and our group is focused more on the B and T cells. There's this companion paper by Mike Beck's group that's also looking at the non-TMB cells, looking at the other leukocytes. But looking at our gating strategy, this is gated on lymphocytes, gated on size and singulates and live. But from there, we can look at CD3 by CD19. From the CD3 gate, we can identify CD8 and CD4 T cells. And from the CD19 gate, we're going to identify non-B, non-T cells and B cells. And so looking at the population frequencies, what we see is that there is a slight decrease in B cells in disease severity. And again, in COVID-19 patients, I apologize, COVID-19 is in red, recovered is in blue, and healthy donors in green. We do see a proportional increase in non-B and non-T cells in COVID-19 patients. And then consistent with what we see with the lymphopenia, we see a drastic loss of T cells in COVID-19 patients. Now, looking at these T cells broken down by CD4 and CD8, we certainly see a loss of CD4 T cells and a loss of CD8 T cells in COVID-19. But when we look at the ratio of CD4 to CD8, we do see an increase. And this tells us that and I would highlight the graph farthest on the right as where the vertical line represents clinical lymphopenia. And so as patients come closer to lymphopenia, there's an increase in that CD4, CD8 ratio, suggesting that there's this lymphopenia is associated more with the CD8 T cell loss, which is slightly unusual, but is consistent with some other groups have reported with loss of CD8 T cells. Now we can further start looking at the different T cells and look at the different phenotypes and B cells. And so we're going to first focus on the CD8 T cells. And I think every immunologist has their different ways of gating, but for the most part, you can tease apart CD8 T cell populations by different expression of CD27, CD45RA, CCR7, and CD95, and we can use these gates to tease apart naive central memory effector memories 1, 2, and 3, and effector memory cells that express 45RA, or MRES for short. Now, looking at these cells, we definitely see changes in these different populations where there is a slight loss of naive CD8 T cells in COVID-19, as well as losses in EM1s. But we do see increases in EM2s and these TEMRAs. When we start looking at the phenotype of these cells, well, we don't see any changes in PD1. We do see increases in CD39 in patients with COVID-19, and CD39 is a marker that is associated with the exhausted cells. Although it's interesting that PD1, which also is associated with exhausted cells, isn't up. So in summary, we do see increases in decreases in EM1 and increases in EM2 and MRES. We do see CD39 increase in non-naive CD8 T cells, and I didn't show you here, but PD1 is increased in central memory and effector memory 1 cells. But one of the take-home themes that we're going to see throughout this paper is that there's incredible variability in the phenotype of these patients. So when we look at, for example, activation as measured by CD38 and HLA-DR, we see minimal activation in healthy donors, recovered donors. But in COVID-19, we see patients who either have this really activated phenotype or very minimal. And that can be enumerated on the far right, where this green line essentially captures, this green horizontal line essentially captures where the healthy and recovered donors lie. And we do see a population of patients either above or at that line, suggesting that there is this broad heterogeneity in COVID-19, at least in the CD8 response thus far. A similar phenomenon is true in Ki-67, where we do see minimal activation in healthy donors and covered donors, and certainly that's the case in COVID-19 in some COVID-19 patients, but not at all, as this patient's clearly presenting with 11% frequency of Ki-67, they're non-naive CD8s, which is very unusual. And if you, again, looking at the enumeration, you can clearly see here that patients have a very high proliferative response in their CD8 compartment, although a lot of patients don't. So this tells us that so far in the CD8 T cells, we see this variability in both population dynamics as well as phenotype. Now we can take all this data and project it onto T space, where we can see different regions highlighted in healthy donors, recovered donors, COVID-19. Well, the take home here is that you see very similar areas lit up in the healthy donors and the recovered donors. But in the COVID-19 patients, you see areas that are lit up that are not the case in healthy and recovered donors. We can start to identify what the phenotype of those different regions are. And so if we look at, for example, TBET or CX3CR1, what we do see is regions that are lit up that are TBET positive in COVID-19 patients that are not as apparent, I should say, in recovered and healthy donors. And certainly that's the case with CD95, where CD95 being fast expression, and that the region that's identified as fast positive is very lit up in COVID-19 patients. We can cluster this data into 10 or 15 different groups, and each group having its own unique cluster. And what we've noticed is that clusters 11, 14, and 15 are identified as kind of the responding cells as measured by Ki-67. And we can identify the phenotype of those cells, and we can take those clusters and project them back onto the to biaxial plots to look at the phenotype of those cells. And we can see, and what I should say is that these clusters are associated with changes in COVID-19, as cluster 11, 14 cluster 11 is significantly decreased, and 14 is increased in COVID-19 patients. So all in all, what this tells us that so far in the CD8 compartments, there's considerable heterogeneity, that we see differences in populations, we see differences in phenotype, and at least in the CD8 T cells. But how does that clinical change, how does those frequency changes relate to clinical parameters? And now we can take those CD8, those populations and relate them to the different clinical features. And some interesting phenomena will occur, some that you'd expect. And the only one that had a false discovery rate of below 0.5 is that naive cells negatively correlate with age, as you'd expect, as indicated here. But some other unique features came up. For example, that the activated CD T cells positively correlates with blood donor type, and this is a part of so how blood types correlate with SARS-CoV-2 infections are coming out. Still more evidence needs to be done, but we're certainly seeing differences in immune features with blood type, which is interesting. We do see some other things you'd expect, as Ki-67 positive correlates with IL-6, and there's, again, this plot really identifies a lot of different relationships that can tease apart how the CD8 T cells relate to different clinical metadata features. Now we can do a similar type of analysis in the CD4 T cell response, where, like the CD8 T cells, CD4 T cells can be split apart by different expressions in CD27, CD45 RA, and CCR7. And we can also look at TFH cells as measured by CXCR5, PD1, and we can look at the activation of those circulating TFH cells as double positive of CD38 and ICOS. And so I'll just project all the data again. And so, like we saw in the CD8 T cells, we do see considerable heterogeneity in the CD4 compartment as well. If we look at Ki-67, we certainly see an increase, a population of patients in COVID-19 that have an increase in Ki-67 than non-naive CD4s. But as the similar green line, which encapsulates the healthy donors, there's a lot of patients below that line. And similar phenomena is true with activated cells. And similarly, it's like a similar analysis to the CD8 compartment, we can project this data into t-SNE space, and we definitely see different regions lit up in COVID-19 patients that are not apparent in recovered and healthy donors. We can also break this data up into different clusters, and 15 clusters in total, and we highlight clusters 14 and 15, which are uniquely lit up, the bottom region here, which are uniquely lit up in COVID-19 patients. And we can look and see what are the flow features, what are the phenotype of those clusters. And so those clusters can be identified as responding cells as measured by Ki-67, or some type of effector memory or effector cell as measured by CX30CR1 at TBET. And we do see those clusters lit up, or increase, I should say, in COVID-19 patients. And again, similar to what we did with the CD8 T cell compartment, we can take those these CD4 populations and ask how they are related to the clinical metadata. And similarly, and so some interesting correlations come out. The naive T cell compartment negatively correlates with PCT, PCT being another metric of inflammation in patients. We certainly see positive correlations of CD4, Ki-67, and 38 with ferritin levels, ferritin being a marker that's been pretty consistently reported on being elevated in COVID-19. But what's interesting is that we see activated CD4 T cells positively correlating with the Apache score. Now, the Apache score is kind of a summation, a numeric evaluator of organ failure in patients. And so we definitely see a lot of activated CD4 cells associated with clinical metadata that suggests organ failure or inflammation, which is pretty interesting. And again, there are a lot of other correlations to be made here, but I just talked about a few that were just somewhat interesting. So next we can ask ourselves, well, we see difference in CD4s and definitely see difference in CD8s. So what is the relationship of the CD4s and the CD8 T cells? And so we can do that by looking at non-naive CD4s, looking at Ki-67 and CD4s and CD8s, or activation on CD4s and CD8s. And if you essentially draw a diagonal line across these plots, what you'll notice is that there are patients who have have a coordinated activation of both T cell arms, although there seems to be more of a tendency to have a stronger activation of the CD4 cells. But what's also interesting is you see a lot of patients who essentially cluster very similar to the healthy and recovered donors. And so similar to what we see when we looked at the CD4 cells and CD8 T cells, we see a bunch of patients who have this high activation, but we also see patients with no activation at all, suggesting of this pretty vast heterogeneity associated with this disease. Now, this lack of response towards the bottom isn't due to some type of intrinsic factors, as if you take the patients that have low Ki-67 and stimulate them with anti-CD3 or anti-CD20, these polyclonal activators, they do make interferon gamma upon signal, and this tells us that there must be some type of intrinsic factors that are preventing them from getting activated. Now, I would note that we were limited by cell number here, patient numbers here, only because due to biosafety concerns of virus in the blood, we were not allowed to stimulate a lot of patients unless they were cleared to be virus negative in the blood. So we can now take this similar analysis strategy and look at B cells as well. And so we can break down the non-plasmatized B cells by IgD and CD27 to look at naive and switch to non-switch memories. And we definitely see decreases in both those memory compartments of COVID-19. Now, I think one of the most striking things was the increase in plasma cells. And we are calling plasma cells at CD27, CD38 bright. And what you'll notice is that in some patients, it can reach as high as 30 to 40% of the circulating B cells. Now, this is very unusual. In cases of influenza, it perhaps reaches around 10 to 15% on the high side. And this type of response matches closely to more like Ebola or dengue, which is obviously not coronaviruses. So why it's this high, we're still trying to determine, but it's really, really interesting. But like the CD4s and like the CD T cells, you see a lot of patients who have an increase in a pretty substantial increase in plasma cells. We also see patients who have very similar responses to healthy donors. And like what we did with the T cells, we can now project these patients to T space and to identify the clusters that are lit up in the COVID-19 patients. And what we can identify is the cluster that's the top right is probably your plasma cells, as indicated by CD138 expression. And so we can just focus on the clusters that are highlighted in COVID-19, and we should identify those to be cluster 1 and cluster 5. And what we'll see is that these clusters, the phenotype of these clusters as looked at by TBET and CD20 or TCF expression, and cluster 5 being the plasma cells, and as you'd expect, they're much higher in the COVID-19 patients. And we can now, like a recurring theme, we can look at these different B cell populations and see how they relate to the clinical metadata. And the strongest correlation, as it's the only one that had a false discovery rate of less than 0.05, was the naive B cells being positively correlated with lymphocyte Now, what was interesting, although it wasn't, I think one of the most interesting findings was that the plasma cells did negatively correlated with IL-6. Now, I think in a lot of these patients, we increased IL-6, and our thoughts going into this was perhaps it's the IL-6, IL-21 axes that's driving this plasma cell activation, but that does not necessarily be the case here. So what's driving, we're still kind of determining that, but it doesn't appear to be IL-6 at this point. And again, a lot of other trends are emerged where you can relate these B cell populations to the clinical metadata. But then one of the questions, and I would highlight, this is the work from our companion paper by my groups, by Letty and Bettina in Science that was also published, where they looked at the B cell repertoire. And how to interpret this plot is essentially the more yellow and gray you see, the more polyclonal response, and the more blue you see, the more monoclonal response, or the reduction in the repertoire. Oh, I'm sorry, I said it backwards. I'm sorry. The more blue you see, the more polyclonal. The more yellow and gray you see, the more oligoclonal. And what the take-home is that patients that are, and the squares are the healthy donors. And so the take-home of this is that patients that have disease, you see more orange and more yellow, suggesting that there's more of a restricted clonality in COVID-19 patients. And now, certainly, if you look at the D20 score, and the D20 score is a scoring of the top 20 repertoires over the total repertoire. So the higher the number, the more, the less of repertoire these B cells present with. And what you see is that when you have a high plasmablast, there's less of a repertoire, suggesting that this appears to be some type of antigen-activated response. And that is further confirmed by looking at the CDR3 links, where in severe patients, they have an increased CDR3 link. So the summation of all this data is that while we certainly see this activated plasma cell response, it does seem to be antigen-driven. To what antigen? There's still a lot of outstanding questions here, but it's certainly an interesting phenomenon. Now we can then ask, how are the B cells or the antibody response related to the other lymphocytes, be it plasma cells or TFH cells? And there doesn't seem to be a relationship between the plasmablasts and the anti-SARS-CoV-2 IgG that we're looking at. Now, I would note that this is looking at the RBD region, and so it could be that the plasma cells are responding to other determinants of the virus. We don't see much of I mean, it's a little bit more, but it really isn't that significantly the correlation between plasmablasts and activated circulating TFH. but there is a relationship between the activated TFH cells and anti-SARS-CoV-2 IgM and antibody levels, being IgM or IgG that are SARS-CoV-2 specific. This is consistent with what we see with other groups who have reported that IgG titers are correlated with the activation of the CD4 cells, as reported by Alexander Sedes and Shane Carty's group a few months ago. So I think one of the common things are what the cytokine levels are, and so while we're certainly limited by patient number, we were able to determine some cytokines that are up. CXCL9, CXCL10, which are classic interferon-driven chemokines, they are increased in COVID-19 patients. So is CCL2, perhaps some type of monocyte response. But IL-6 is a common one we're asked about, and IL-6 in this study did not appear to be up, although in the clinical data it did. Now, why could this be the case? Well, we believe that the patients that we selected were randomly selected from our cohort, where in the clinic, they typically do the IL-6 measurements on patients that they suspect have high inflammation. So it's essentially biasing the selection to patients with high inflammation where you would expect higher IL-6 levels. But there is mixed reports on IL-6, and we see definitely both in our data set. So we're still determining what is going on with IL-6 in these patients is work we're currently doing. Now, a common question they're also asked is, how does this change over time? And so because some of these patients stay in the clinic past a week, we're able to collect longitudinal data. And so when we project that data into kidney space, the take home from this is that you see the same regions certainly lit up. And what I should say, these are what's lit up are the COVID patients. And what this tells us that the immunological response is surprisingly relatively stable over 7 days. And this is not what we expected. Now, we can certainly do some bias gating and look at different populations, be it Ki-67 or HLA-DR on CD4, CD8 cells, and we do see changes accordingly, but on a kind of a global scale, it remains fairly stable. So what we can do is we can now take patients that have, we're going to, for the next study, we decided to group patients into patients who have increases in Ki-67 or patients who remain stable. Now, I would note there was no patients who had a loss of Ki-67 in over 7 days, but that wasn't the case in plasma cells, where in certain patients we see it go up over time. In certain patients, the plasma cells remain stable over 7 days, and I would note that this patient remained stable with a plasma blast frequency of 30%, which is remarkable. And in some patients, we see it go down. And so what we can do is we can take these patients and break them up into those different groups and ask, how does that relate to that clinical metadata? Now, what comes out is some interesting observations. And so, for example, I'll just talk you through the top one. It's really interesting, is that when we see patients whose activated T cells go up in the 7 days, what you'll notice is that at day 0, they don't really present with any humoral antibody response. But over the 7 days, and this is looking at the full change from day 7 to day 0, they do make a response. Okay, whereas patients who come in with really their Ki-67 responses remain stable over the 7 days, well, they don't really, they have high levels already, but it doesn't appear to be going up. And so what we believe what we're capturing is a snapshot of time. So what this is that when the patients are going up, these are patients who are just mounting that immune response. Whereas if they remain stable, what we're thinking is that these are patients who had a pre-existing response or in the midst of response. And so this obviously has some implications with therapeutic strategies of what, and from the previous studies, we talked about the timing of interferon lambda is important. And I think this type of data analysis tells us that the timing does have differences in the clinical metadata presentation, which is fascinating. And so, I mean, there's a lot to talk about here, but for the sake of time, I'll just quickly pass over this because I want to get to the last point, which is severity. And so what we talked about at this point is how the COVID data, how patients present themselves with COVID-19, and we talk about the changes over time. But obviously, one of the bigger things is how the disease severity. And so this is based off the NIH ordinal scale, where COVID-1 is death, but from COVID-6 to COVID-2, you require hospitalization, whereas COVID-7 and COVID-8, you don't. And so the biggest punchline is that T cell frequencies decrease as a function of severity. So the worse disease, the less T cells you have. And certainly we can look at the phenotype, but again, for the sake of time, I just want to get to the last point. And you can take all this data and you can project it on a heat map, and you certainly see distinct clusters emerge. But the problem with this type of analysis, you have kind of biased lymphocyte gating. And so the question is, how do you take all this lymphocyte data and project it onto space so you're not kind of using biased populations? And so what we decided to do was project all 193 immune features into a UMAP space, and then look at how this population changed in relation to disease severity. And an interesting pattern emerges. And what you'll notice is that, and I want to highlight that only immune features was used to build this heat map. You build this UMAP. And then how the patient resolves themselves around the x-axis or component 1 is by disease severity. And so what you'll notice on the far left, you have the healthy donors, but as you go along this x-axis or component 1, these patients get worse and worse in terms of disease. And that for us is really interesting because disease severity wasn't used to build this UMAP. And so now you can look at different immune features and say, how do they contribute to the spatial alignment of these patients in this UMAP space? For something like activated CD4 cells, they kind of have influence in both the component 1, the x-axis, component 2, the y-axis. But something like T-beta expression, CD8 generally drives their expression along the y-axis, whereas plasma cells drive their expression along the x-axis. So we can now take this data, and I'll skip this top part, and I'll just focus here for now. And so we can take those different axes and say, how do they line themselves up with the clinical metadata? Okay, and what we see is that component 1, as shown by the patient data, aligns itself with disease severity, as a lot of these clinical parameters that are associated with worse prognosis or disease severity are positively correlated with component 1, which is not the case in component 2. So if I go back to this axis just to kind of further explain that point, is that a lot of those like Apache score, kidney injury, all these things are associated with along this axis as opposed to component 2, which is interesting. And so from this, we can now define different patterns of immune cells that emerge. And so what we define this as is patients that have high component 2 but low component 1, or high component 1 and low component 2. And that is what we're defining as immunotype 1, which is typically patients that have high component 1, or immunotype 2, which are patients who have high component 2 and low component 1. And they definitely have differences in immune features. And again, for the sake of time, I'll just go over this because I want to talk about the last feature, which is there are patients who have low component 1 and low component 2. And these are what we're calling immunotype 3. And they're defined as these patients who have responses similar to healthy and recovered donors. So overall, we're defining these 3 distinct, what we're calling immunotypes, as a way to kind of bend the different responses we see in COVID-19. And in the paper itself, it defines how these different immunotypes associated with disease severity. And I apologize as I'm trying to skip time, so I'll just summarize real quick, is that what we found in our data is that there's a considerable heterogeneity in the responses. There's definitely some CD T cell bias, and why this is, we're not really sure, but we're certainly investigating. And we definitely see a massive plasma response that rivals that of Ebola and dengue. When we look at the longitudinal data, it remains fairly stable over 7 days. Now, certainly if you do some bias gating, you can see population differences, but overall, it remains fairly stable, which is pretty unusual for an acute viral infection. And then we can now take all that data and project into UMAP space and kind of bin these 3 different immune responses as immunotype 1, 2, and 3. And each one has a different immune features that define them. And we believe that these different immunotypes will have implications with treatments, whether it be immunotype 3, which has low activation, might need some type of immune stimulatory agent, or immunotype 1, which has high activation, might need some type of immune suppressive agent. This is kind of the work we're doing right now, but I just want to end with, this is a considerable effort. There was a ton of patients that came in a short period of time, and a lot of this work was done with the other first authors who were just amazing to work with. And also I wanted to talk, just the lab itself is wonderful. John's a great mentor. He supports us in every way possible. With that, I'll take any questions, and I apologize I ran a little over.
Thank you very much, Divij. It's really a fantastic study. It's really impressive the level of understanding you can get from like a multi-dimensional analysis with like an unbiased approach, and you end up like really by defining the phenotype. It's really impressive. So Quen, tell me that we have like time for 2 questions. Okay. So Raj Datani is asking, what are the implications for treatment specifically? Can you say more on the work you are doing now? And maybe I will also like use this to ask also a question about the temporal changes that you present, and that could be really useful to inform of which patient would benefit from interferon treatment versus the one that would benefit from inhibition of interferon pathway. What would you think would be like the best marker from your data to select which patients would be in the early stage of the disease versus late?
Yeah, so I can, let me work backwards on that one because it'll inform that question. So we built that UMAP space on just the flow immune feature. So what we're doing right now is we're going to take this to 10x sequencing to further bring clarity to what defines patients in that UMAP space. Because, for example, immunotype 3 is simply the lack of immunotype 1 and the lack of immunotype 2. And so it's not really a meaningful definition at this point. So we're trying to bring further clarity to this, and that we believe will have implications on what treatment options they could give. For example, to be consistent with the other speakers today, one of the things we're interested in is interferon responses. So which one of these immunotypes is associated with a higher IgG representation in their immune cells, and that will have definitely implications on which therapy we should consider. So we're essentially trying to further define that space, and that will then allow us to solidify what treatment options would be best.
And so can you tell us a little bit more about what are these patients in the immunophenotype 3? Like what are...
Yeah, so at this point, I mean, we just finished the 10x sequencing. So if you ask me this of patients in immunotype 3, and so I was planning to go look at this actually today itself. So I can certainly give further definition on in the later weeks to come. However, at this point, it's simply that green line that was drawn in regards to Ki-67 or active and HLA DR and CD38 that was similar to healthy donors, recovered donors. That was essentially the only parameter upon selection for immunotype 3. And so we are now working to see what other features could define it. And so it's not, again, it's not the most meaningful definition at this point, but it had to be done to kind of define that space in the UMAP.
All right. There is a question from Brian Bernal. Great work, Divij. I found really interesting the culture supernatant experiment. I would like to ask if instead of the anti-CD3, CD28 stimulation, you use a SARS-CoV-2 protein like spike or other protein for stimulation, you expect to have the same result, since patients won't have polyclonal response, but something that would be more specific.
Yeah, so we are doing this currently right now. I'll say the one limitation we have is we are still, by our EHA standards, can't stimulate COVID patients. We can only do recovered at this point. So we're working actively to see how we can stimulate these COVID PBMCs, because I think that'd be very interesting. So now if I were to talk, so we are doing that exact experiment, and we're using the megapools as developed by Alexander Sede and Shane Crotty. And we are seeing similar to what they're seeing. We are seeing more of an activation in the CD4 responses, and that's consistent with our flow data. I haven't seen to this point, but I've only done a few patients, is that they have seen responses in the non-spike region in healthy donors, suggesting of pre-existing immunity. We haven't seen that yet, but I think that's really, really interesting, as it has implications of pre-existing immunity is causing this immune heterogeneity. So stay tuned, because that's definitely what we're doing.
All right, Quan, should we?
Yeah, I think the time is up, and I have so many questions, but thanks to all the speakers for the great talk. Thanks to Jack, thanks to Achille, and thanks to Divij. And thank you very much, Nicholas, for hosting and all your exciting ongoing work on COVID-19. I think it's crazy to think about how half a year ago there's no COVID science. And I think, you know, although the world is still in pandemic now, at least on the science side, I think we're going to make a lot of great progress. So in the end, I think we can take one group shot for whoever wants to come and show their faces. We can take a group shot. In the meantime, I also want to advertise, we also have a neuroinflammation symposium tomorrow at noon Eastern time. So Jerry is sharing the information for tomorrow. You can see it on our website for the information. And so I think that's it. I think there's probably a lot more questions. We'll post the video on YouTube, and you can go there and discuss more. And thanks to everyone. The speaker also can leave the information if you have additional questions. And so thank you, everyone. I think that's it. Thank you.
Thank you so much for inviting us.